Hello and welcome to another video in our course on cell biology and genetics. In this video, we are going to talk about transcription. So what are you going to learn in this video? First, I'm going to explain what transcription and translation are, and we are going to look at the process of transcription in detail. We are also going to talk about post-transcriptional processing and introns and exons. I am also going to explain what alternative splicing is. After watching this video, you will also understand the terms transcription bubble, transcription factors, promoter, TATA box, transcription initiation complex, terminator, capping, polyadenylation, splicing, pre-mRNA, snRNA, snRNP, and spliceosome. By the way, you can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. Transcription is the first step of gene expression, the process in which the information in the gene is used to synthesize a product, usually a protein. Transcription is the synthesis of RNA from DNA, or in other words, the transcribing, rewriting of the DNA code into RNA. The second step is then translation, when a protein is synthesized from this RNA. Transcription begins with the unwinding of the two DNA strands by RNA polymerase. The structure we get by unwinding the DNA strands is called a transcription bubble. One strand then acts as a template for the synthesis of the RNA. The RNA is synthesized by RNA polymerase that adds new ribonucleotides following the basic base pairing rules, with one exception. RNA doesn't contain thymine, but a different base called uracil, which also pairs with adenine. As was the case with DNA polymerase, when we talked about DNA replication, RNA polymerase can add new nucleotides in the 5' to 3' direction only. For that reason, the template it uses always runs in the 3' to 5' direction. Unlike DNA polymerase, RNA polymerase can start an RNA strand without a primer. However, RNA polymerase needs accessory proteins that we call transcription factors in order to start transcribing. These transcription factors bind to a region preceding the gene that we call promoter. This promoter often contains a sequence of thymine and adenine nucleotides, the so-called TATA box. The transcription factors, together with RNA polymerase, form a transcription initiation complex, and then transcription can start. RNA polymerase synthesizes the RNA until it reaches a certain sequence called terminator, where it stops and releases the new RNA. Because it transcribed only one DNA strand, the RNA is single-stranded. Now, usually genes specify the amino acid sequences of proteins. Simply said, these genes tell us what amino acids are going to be present in a certain protein. The RNA molecules we get from transcribing these genes are called messenger RNAs, mRNAs. These genes are transcribed by a special type of RNA polymerase II. We can, however, get RNA molecules as the final product of the transcription as well. So again, instead of protein as our final product, we get an RNA molecule. These include specific RNAs that we call non-messenger RNAs. For example, rRNA, tRNA, etc. We are going to talk about them in the future. These RNAs are transcribed by RNA polymerase 1 and RNA polymerase 3. In eukaryotes, transcription takes place in the nucleus because the DNA is stored there. However, translation, the process during which a protein is synthesized, takes place in ribosomes in the cytoplasm. For that reason, the RNA must be transported there. However, the RNA must first undergo the so-called post-transcriptional processing. So in other words, processing after transcription. And there are three processes that need to happen. The first one is called capping. RNA capping is a process in which a guanine nucleotide bearing a methyl group is attached to the 5' end of the RNA. Then we have polyadenylation, which is a process in which a series of adenine nucleotides is added to the 3' end of the RNA molecule. Because multiple adenine nucleotides are added, the process is called polyadenylation. Poly means many. Capping and polyadenylation are important because they protect the RNA molecule from degradation, facilitate its transport from the nucleus to the cytoplasm, and facilitate translation. The third process is called splicing. Splicing is a process which needs to happen because of a special structure of eukaryotic protein coding genes. These genes contain two important sequences that we call introns and exons. Introns are non-coding sequences. In other words, sequences that do not contain instructions on how to make a protein. Exons, on the other hand, are coding sequences containing these instructions. As introns do not code for proteins, they need to be removed, and this is done during the process of RNA splicing. During this process, introns are removed from the RNA molecules and exons are put together. RNA splicing is carried out by special RNA molecules called small nuclear RNAs, snRNAs, that together with proteins called small nuclear ribonucleoproteins, snRNPs, often pronounced as SNRPs, form a special structure called spliceosome that is responsible for the process. Before post-transcriptional processing, the RNA molecule that was transcribed is referred to as pre-mRNA. After capping, polyadenylation and splicing, the RNA is ready to leave the nucleus and is referred to as the mRNA, messenger RNA. What is interesting is that the transcripts of eukaryotic genes can be spliced in many different ways, producing different proteins. And this is called alternative splicing. So again, alternative splicing is that genes can be spliced in different ways, and this is going to produce different proteins. Okay, so now we'll summarize what we've learned today. First, we said that transcription is the rewriting of the information stored in the DNA to an RNA molecule. Translation is then synthesizing a protein based on this RNA molecule. We explained the whole process and said that it starts with the RNA polymerase, which unwinds the two DNA strands and synthesizes the new RNA strand in the 5' to 3' direction. And it doesn't require a primer. However, it needs transcription factors, which bind to a promoter, and together with RNA polymerase, they form a transcription initiation complex, and transcription can start. RNA polymerase then synthesizes the new RNA molecule until it reaches the terminator. We also said that we have different types of RNA polymerase. There's RNA polymerase II, which transcribes genes that specify the amino acid sequences of proteins, and the product of transcribing these genes is an mRNA. Then there is RNA polymerase I and RNA polymerase III that transcribe genes that code for non-messenger RNAs. So, for example, rRNA, tRNA, etc. We also talked about post-transcriptional processing, so capping, polyadenylation, and splicing that needs to happen before the RNA molecule leaves the nucleus. During splicing, the non-coding sequences called introns are removed and the coding sequences called exons remain. Finally, we said that the RNA molecule before post-transcriptional processing is called pre-mRNA, and after it is called mRNA. Okay? Great, so this is it for transcription. Thank you so much for watching. If you found this video helpful and you would like to watch more videos on cell biology or medicine in general, please subscribe. Thank you again, and see you soon.